Good morning.
Assalamu alaikum and good morning. Professor Iqbal Chaudhary, Director of the ISNPC-15, yes, these distinguished professors, visitors, and folks. Can everybody hear me now? Thank you. Welcome to the exciting session of the last day of this wonderful meeting. We will be starting our seminars with Professor Christian Bechtel from the University of Hamburg Institute of Biochemistry and Molecular Biology, Laboratory for Structural Biology of Infectious Inflammation. Professor Bechtel is a very distinguished faculty member under the Global Initiative, Foreign Fellow of the Pakistan Academy of Sciences, and he has been a distinguished professor and board member of many organizations. Without taking any further, I will invite Professor Bechtel to start his talk on the future prospects in structure-based drug discovery to treat infectious diseases and examples. Professor Bechtel.
Dear Chairman, thank you for the introduction, and I hope you can see my slides and you can hear me well. I'm saying good morning from Hamburg, Germany. You see the title of my presentation on display, and I structured it. I will start with an introduction, introducing new and upcoming methods in X-ray structure analysis. As I'm on the DESY campus, we are involved in many developments, in hardware developments. And the second part of my presentation will focus towards drug and lead discovery in infection biology, focusing on antibiotic design as shown here. This is the bacteria Staphylococcus aureus and our contribution to COVID-19 research. And this is a collaboration also with the Institute in Karachi. Okay, so just to I'm now in Hamburg. This is in North Germany. So Hamburg is a nice harbor city, as you see here, the center of the town and the harbor. However, important for life science research is the storage ring Petra 3 on the DESY campus, the German synchrotron facility shown here in two landscape figures. I'm just sitting here. You see my cursor. This is our laboratory on the DESY campus. And we are utilizing the high-intensive synchrotron radiation for structure analysis. And in parallel in Hamburg, we have the Free-Electron Laser here, just shown. You see here the DESY campus, just here on the left side, the harbor in the background. And here in the scheme, the Free-Electron Laser, which is 3.8 kilometers long, but is underground. And I will come back to this later on. So our working horse, as you see here on this slide, is X-ray crystallography. And I remember some speakers talked also about X-ray crystallography on Wednesday on this symposium in Karachi. And you see here a figure from the textbook of Bernard Rupp showing the cycle of structure analysis and lead discovery. You prepare complexes of target molecules, mostly enzymes, with potential lead compounds and refine the structure of the leads accordingly. You see here the entries in the protein data bank, which is for me the mother of the data banks, is increasing or has increased exponentially in the last years, no doubt, because many methods also in molecular biology, biophysics, and radiation sources improved substantially. However, as you see here again, the increase in entries in the protein data bank. Last year, in the middle of the summer, the new software tool AlphaFold came up. And this is tremendous also for me and other people working in structural biology. It is producing high accurate protein structures, and the prediction is tremendous. We tried out on many structures and we are super impressed. And this arrow here is indicating just on the spot almost not in one day, but very short notice, 200,000 structures predicted by AlphaFold were showing up. And this changed dramatically the world in structural biology. So you see associated articles here, great expectations, and one, for example, the area of crystallography ends. And I made this picture here. So this is changing structural biology tremendously. We had presentations here about AlphaFold 2, which is today the main tool of the DeepMind, is the company which is developed or which develops these tools in terms of an international efforts. And in Europe, the EMBL outstation in UK is not responsible, but taking care about this type of software and advertising and also training. So as the name is indicating, this is AlphaFold 2. Certainly the versions 3 and 4 are coming up. This is based once you are placing a sequence of the protein, you would like to see the structure, and the program suite goes into the direction. It is screening the structure databases, mainly the protein data bank, and in parallel is comparing sequences from different sources, human, mammalians, up to plants, and then combining this information. And at the end, you have a prediction of, I would say today, of a highly confident prediction of a protein structure. This question came up last year, and there was a lot of discussion internationally in the community. Do we still need experimental methods to look at structures? And seeing first the performance of AlphaFold is much better than all competitors like ITASA and programs used so far for protein structure or biomolecule prediction. It has tremendous accuracy if you, this prediction with this RMS standard deviation in the range of less than 1 angstrom compared with till now known other methods. As shown here, so the prediction is quite good, but remaining limitations are, which is important to mention, it goes only for single chain type of biomolecular molecules. And we know we have many assemblies, they're showing up as dimer, tetramers, and so on, to be active, and I will show later on examples. Disordered and unstructured regions, similar to experimental methods, cannot be predicted so well. No dynamics, no prediction of mutations, and non-protein components are also not predicted till now, I would say again. As for example, RNA and DNA and protein complexes, metal ions, ligands, for example, drugs, cannot be predicted now, but I understood the next version is particularly focusing on coordination of ions. And this is a summary prepared in the same, so it was published in Nature last year, so the announcement and the launching of the program, and this is an article from Patrick Kramer just saying it's a tremendous program, structures. It provides information for structure analysis and phasing, so we are using this prediction tool also to perform phasing of data we collected, but we could not obtain heavy atoms till now. So we solved a bunch of structures which were pending using AlphaFold 2 prediction, or the structure of Alpha 2 predicted. And particularly in drug discovery, this is changing tremendously current situation. To finish this subtopic here, I can recommend if you go to this webpage, this is the EMBL outstation in UK. They're providing almost continuously a type of webinars and workshops to introduce this type of software to the international community working in structural biology. So now the question is, where is the future of X-ray crystallography? For me, my working horse for many years to solve particular questions in life sciences is, of course, drug discovery, because AlphaFold now cannot predict binding of potential lead compounds, fragments, or drugs. And important is in future the molecular time domains, On the left side, you see again the standard figure of X-ray crystal showing the scheme of X-ray crystallography, crystal, diffraction, Bragg's law, computing, electron density, and interpretation. But independent for the last, I would say, 5, 6, 8, 7 years, crystallography tries to just obtain also information about time-resolved events in structures, as shown here on the movie below. I guess you can see my cursor. This is a penicillin binding protein just assembling a bacterial membrane, and you see a number of movements and domains switching along the active function of the enzyme. And this is what we want to understand in more detail these days, because most of the time we have only static figures about biomolecules. And you see here's a movie from 1878 from Murbright, was the photographer, asked by Stanford, the founder of the Stanford University, to prepare the first movie. So for me and other scientists, this is the first time-resolved movie on a microscopic scale. And to mention Linus Pauling, already he postulated the key lock system, and already during his time he predicted that the principle is shown here. So you have an active site and you have a drug which is blocking or functioning in the active site. However, the lock is not static. So this is the induced type of fit, this type of postulation, and he was already mentioning that the lock is not rigid and we need to understand more about flexibility of molecules. Here you see static figures, you have a ligand, a receptor, and the binding. Coming back to X-ray crystallography, on the top here, you see again the figure showing the scheme, the single crystals. But along the last years and the upcoming, the new radiation sources, new methods were developed. This is shown here on a diagonal. You have now the option to spray crystal suspensions on a tape. We call this tape drive here, developed here on the DESY campus. And you expose with X-rays and you can trigger the sample inside. You can add, for example, a ligand and collecting data sets along the tape movement from many crystals. You can observe structures from intermediates. Another method developed for starting, say, 6, 7 years ago was a liquid jet here. You introduce crystal suspensions via liquid jet shown here and on the right side. And you can trigger events by, for example, photoactivation, also mixing, and you can obtain structures. Today we are collecting data sets in the range of seconds or below from many crystals, and you can follow up intermediates. And I will show you a nice example from a beta-lactamase later on. And last to mention here is the ship development. We have ships with wells, and these ships can have up to 20,000 microwells. You bring in your protein crystal suspension, and by rotating this ship slightly, I have one in front of me, which is a larger ship. I just show a little bit to the camera here. You can collect many data sets at different structural events to look at structures and different conformations to complement. This is a figure you see here from an article prepared by Petra Fromme, one of the pioneers of X-ray free-electron laser application. X-ray free-electron lasers, as shown here on the right, are tremendously intensive. And in Hamburg, this facility is underground. It has 3.8 kilometers. At the end, you have stations to collect data. So they're extremely high and short pulses in the range of femtoseconds. And she compared to synchrotrons at this time. So you have many stations around. And at this time, she was saying relatively long pulses, low brightness. This changed. We are approaching the 4th generation of synchrotron radiation sources. And you can perform also time-resolved experiments as shown here, triggering from in the millisecond range. Again, here, detector sample, and you can trigger even the detector to catch up data sets at different time points. And we are lucky today, just this scheme of the world, and we have a bunch of very good synchrotrons around. Sesame is coming up close to Pakistan, and Pakistan is collaborating with this synchrotron. And here are the Free-Electron Lasers. Sorry, the resolution is not so good. But they're nicely scattered around the world. And I would say the COVID event just was pushing to have more facilities which can collect remote data. So these days, most groups are sending data sets to a facility, and they are fully automatically these days collected. This shows a scheme of the Free-Electron Laser we have here in Hamburg, and you see the building on top and 3 floors in the underground. Here's the tunnel. We have the experimental stations. Here's the building on top. And here are the stations where you, for example, this is Henry Chapman and a co-worker, Anton Barty, very famous these days, collects. This is a station where you spray crystal suspensions and you collect data. And also with this, we can do today mostly remote. I'm changing to the second topic of my presentation. So drug discovery and lead discovery in infection biology, focusing first on antibiotic design and discovery. And it was all the days before, like finding a needle in a haystack. This is today a little bit more advanced. Your screen as shown here from the figure of Bernard Ropp in the textbook. First ligands as shown here, you have compounds, and then you collect data sets, you optimize your compound up to have nice binding constants. And of course, you have to check the cytotoxicity in parallel, so you need microbiology labs nearby which are complementing this. It's important to mention we work with crystals, and you see here 2 crystal or proteins in crystal structures rotating. You need solvent channels that the drug can reach the active site. And we follow these days the rule of Lipinski, just shown here. You need a short number of, for the beginning of H-bond donors, except as rotable. And for me and others, most important, the compounds should be water soluble as possible. And this is a certainly limiting case. Many drugs need DMSO or ethanol to be soluble, and some crystals don't like this so much. So this is important to show, and I'm coming back to this also. In parallel, I just show slides about drug discovery. Here, you know, it can take up to 12, 15 years from the idea, bioinformatic drug design, up to phase 1, 2, 3, and drug registration. And it became very famous in the last year, the drug repurposing, and we heard also in the talk of Professor Chaudhary, drug repurposing is a very important tool these days, is meaning for drug repositioning also is the same. You have today data banks of thousands of already approved drugs, and you can just commercial or via collaborations obtain these data banks in your screen against these drugs, against a new target. The advantage, not necessary to mention, you know the drug safety and is already approved. So if you identify a hit, you can straight use for application for a new type of treatment, and examples will follow. As drug discovery and drug repurposing became very famous also on top of the COVID-19 event, just to mention here, you have 2, 3 very, very established web pages. You can go to the sites as shown here. Drug repurposing hub, you have about SARS Cov cancer, and you can screen what is known, what type of repurposing libraries they recommend, and you can learn many things before starting an experiment. This is another one, and this is the web page, Go Drug Bank. You see, you can search for drugs, and you can screen where they are used for, or what is the application, and do your own bioinformatics and take decisions if you want to go towards repurposing. This is an interesting approach. We followed also in terms of antibiotic design or rediscovery. We are working together with our university hospital here in Hamburg with the microbiology, just to understand more about antibiotic resistance on a structural level. And as you see here, the first discovery, everybody knows we have some shortcoming about new antibiotics these days and drug resistance. And there's a strong need to find new type of drugs, or say antibiotics in the near future. As for example, MRSA is well known as a methicillin-resistant bacteria, also known as a hospital type of infection here. In particular, can catch immune suppressed patients and can cause a lot of burden. And here's a scheme just showing the resistance mechanisms well known. So what bacteria are known, they can change the targets, they modify, they can degrade the drug as beta-lactamases and so on. And we were tracing, or we are also in the field of product development, targeting vital enzymes of bacteria and redesign of novel inhibitors. This is the same scheme, just showing the structures behind the scheme you have shown before. Lactamases are well known. This is an ongoing target, penicillin binding proteins, as well as the ribosome here. And study is such an ongoing by a PhD student is here. So we have a beta-lactamase from bacteria epidermis, and we are looking at the structure and complexes. And here, sorry, I'm speeding a little bit up, don't read everything here. These are the last line antibiotics known for pharmacists and medical doctors. And just upcoming, very interesting, new type of antibiotics. I focus here, they have a Bohr atom on this side. And boronic acid is known for many times in chemistry to inhibit serine proteases. And we followed this up. We were screening one repurposing compound library. We found bortezomib, which is an anti-cancer drug, which we solved the structure in complex. Here you see the electron density. And last year, bortezomib has several substantial side effects. Successor came up, ixazomib, and we again were lucky we could solve the structure of the complex to 1.1 angstrom, so very high resolution. And in the second attempt, just this is going on now to test this anti-cancer drug to treat also severe bacterial infections. That is already approved. And just as a scheme showing, we performed time-resolved experiments using tape drive, liquid jet, and chip. And this movie is starting showing the lactamase, and we added first boronic acid in the active site, and you see time resolved the evolution of a covalent bond in the active site. Just in steps, the electron density is changing. You see here different time points. And this is tremendous. This is a new feature in crystallography. Using fast detectors and pulsed radiation, you can see intermediates and you can catch most variable information to redesign drugs, which we were looking on only static before. You see here the bond formation. And due to the time reason, I'm going to the next slide, just showing a summary. You see here the different time points at the end, the covalent bond between the boronic acid. And we modified, so we had some idea, just it was a type of lab accident. We added glycerol here. Glycerol is known for structural biologists as a cryoprotectant. And again, you see the same procedure. We collected data sets at different time points in the range from milliseconds up to 10 seconds. And you see the bond formation of a cyclic ester bond. And this was tremendous. Also, one solvent was here, it's just moving. And you see here, so this opens complete new ways in drug discovery to find new antibiotics or procedures to block the beta-lactamases, which are known to hydrolyze the penicillin and the beta-lactam. Again, here, just you see the evolution of the electron density here, and this is tremendous. And we are ongoing, so we are checking with microbiologists now if we can combine this type of chemistry to treat really antibiotics, to treat bacterial infection. Just in briefly 2 slides in collaboration with my second home, I would say, in Sao Paulo, the University of Sao Paulo, we are looking at pathways which are vital for bacteria. So bacteria produce the vitamins here, if you're shown on the left side. Humans, you know, we have to uptake. Therefore, the vitamin production pathways in bacteria are very interesting drug targets. And we use a prodrug procedure. Prodrugs were also mentioned along the symposium by a previous speaker, just showing what the prodrug means. So you have a compound which is close to the natural substrate. It is hydrolyzed or is modified by the enzyme, but the product at the end cannot feed the cofactor, and in this case, the pathogen will die. We looked at 2 cycles, the vitamin B1 cycle. Also, I had PhD students from Pakistan were involved in this, so we could don't go too much in detail. There's a number of enzymes producing, using HMP and THZ to produce thiamine, which is vitamin B1. Lucky we could solve all structures. That's Tim here and 10.8 in here. And Tim was our target. So we used to identify prodrugs which are similar to the natural, substrate THZ. You see the structure and complex. We redesigned some compounds which are close to THZ, shown here, known that the sulfur is essential. So we found compounds which are in the structure close to the substrate, a number of compounds, and you see here we found one which is just suppressing the activity of a bacteria, probably because the product is, cannot be handled by the cofactors. And you just submitted a manuscript on this, is the vitamin K2 synthesis pathway. We solved one structure, DHA, which is a thioesterase. It's forming a dimer, very interesting, and you have in this complex, sorry, it's a tetramer, we have 4 active sites, and we found first peptides which are really nice, functional as a type of product as well. Now I'm coming to the last part of my presentation, and I have to speed a little bit up, but you can send me questions afterwards. Along COVID, we joined international efforts to look at targets which are vital for the bacteria for the virus. So you know the strong efforts and successful efforts to identify vaccines. And it's same in parallel drugs. As you saw, the drug development can take up to 12 years. So we and other groups went straight to repurposing. And we also found very nice candidates which are suppressing the replication of the virus. Important to mention, just Pfizer launched Paxlovid, which is a drug which is inhibiting also the main protease of the virus, but it needs a relative high dose of this type of drug. Infection cycle, so you can target the uptake of the virus to the cell or the replication. And here are a number of names. This is an old picture from 20, you know, this chloroquine, hydrochlorine, a lot of things going on at the beginning to identify drugs which can inhibit the main protease, shown here, which is essential, and the PLpro. Both enzymes are essential for and vital for the viral reproduction. And international, and as you see in the protein data bank, many groups spent tremendous efforts to analyze these both structures. The MPro for me is more simple, and the new drug from Pfizer is approaching this, successful approaching this protease, which is shown here as dimer. more interesting at the end is the PLpro, which is also influencing the immune system of humans. I'm coming back to this. So these two proteins are the most interesting targets until now, and will be, of course, forever. And in 2020, in February, when the COVID-19 came up to be, how to say, dramatic, we formed a collaboration here in the Hamburg area. So many institutions joined these efforts for repurposing drug approach. So we obtained 2 libraries, and I'm very thankful we obtained a library of natural compounds, the topic of this presentation, from the Karachi Center, from Professor Chaudhary. And what we did, we crystallized both proteins. The main protease is easy to handle, the PLpro very difficult. But we succeeded protein production and obtaining this compound libraries. One of our collaborators had robots that pipetted this these compounds in Limbro plates and multi-well plates. And we performed co-crystallization here, harvesting. This was Hina Anderlip. She's back to Multan University. At this time, she was in Hamburg as PhD student, collecting data, reprocessing. And at this time, or till now, and maybe forever, everything is remote. So you can even work from home to control robots, control data collection and data collection. So most steps are remote. One step is till now not remote. You meet humans for checking crystals and harvesting. And we harvested almost 7 days, 24 hours with 8 people, 3,600 samples. And then the robot is back, shown here. Samples were shock frozen. This is a beamline, which is almost next to me in another building. And we collected data sets. This is again a movie showing procedures. I have to click forward soon. You have a detector here, you have crystal data handling here, and you collect data sets. Almost in 30 seconds, you have one data set. And we process data from here, Hamburg, up to colleagues in Moscow. They were involved in processing a tremendous number of data sets. Here's a figure showing the group working in front. We had a number of hits in the early. Here you see we were very happy the procedure was working. The compound in the active site is the main protease. Here again, active site is a dimer. The main protease is forming a dimer. Both the PLpro and the main protease are cysteine proteases. And I'm wondering because why evolution selected cysteine proteases. Cysteine is very sensitive to oxidation, but we know the virus is very, and I would say, too effective. And this is a natural substrate of the virus, so it's cutting the main chain of non-structured proteins to be reassembled to form a new virus. And we found a number of covalent binders from the repurposing data bank I'm showing here, also overlapping in the active site of the main protease and in different conformations. Also, we identified an allosteric site, very interesting. This is out, here's the active site. One binder is found here, also in this view. And this is showing you some statistics from the fully automatic data collection as a resolution range, the performance, the quality of the data is shown. And of course, after you need humans again to perform the interpretation. And we found 2 drugs, pelintinib and if I'm pronouncing wrongly, ifenprodil, which are known in their kinase inhibitors. They are somehow in application to treat some tumors and cancers, and we found them nicely bound in the active site. And on this table, you see all hits we obtained from compounds which are already in pharmaceutical application. We deposited almost immediately all data in the protein data bank, so they were freely available to pharmaceutical companies. actually all groups in the COVID research published without placing patents, all this data to be freely available for the community. And of course, we combine them with NMR, SPR, and ITC measurements. And this was one of our first publication in Science, which is till now one of the most cited papers. And here are the authors, and I coordinated this with my colleague, Alke Meens. I'm briefly coming back to the second protein, which is more interesting for me. This is the PLpro, also vital and essential for the enzyme. So we could succeed to crystallize this. And if you look in the PDP, there are hundreds of structures for the main protease, but only a few for PLpro, which is because it's very difficult to handle. Don't be worried about the slides. I'm just going with the cursor along. The PLpro is involved in suppressing the immune response of humans, of the host, because it is also it is hydrolyzing the ESG-15, which is an essential molecule in the immune response of humans. So showing here, ESG-15 is formed. So this is an antigen, in this case, a virus. And if you follow my cursor, it's assembling a longer chain, and which is what is known today. The PLpro can hydrolyze ESG-15 at 2 positions. Again, here SARS Cov PLpro. And this causes a cytokine storm. We know this on one side. So patients hyperreact on this, and they can prolong the inflammatory process and the survival of the virus. I have to say, I suffered myself. I was fully vaccinated in December. I was catched by COVID, by the Delta variant, and I was suffering very long, fully vaccinated. And my colleagues from microbiology followed my titer of the virus, which was still invisible or detected up to almost end of January. This made me more interesting in this enzyme again, because it's modulating the immune system. And we applied a data bank obtained from, you are sitting close to this building I'm showing here, and you heard a lot of natural compounds we obtained from the Karachi Institute, this compound library. We started screening, same procedure with PLpro, and we collected for first 500 compounds data sets. This is a PLpro again, the active site here, and ESG-15 is binding here. This we know today. Before it was not known, but we have structures. And you see electron density of, we have followed up first 3 compounds, which were super interesting. And here again, active site, ESG-15 is binding here. So this enzyme has 2 functions. It's cutting the main chain, the non-structured main chain of the virus, plus modulating the immune system. Again, this is, the compound, whoops. I'm coming back later with a summary on this. And it has a lot of mobility. Now we are going also to perform time-resolved experiments. 2 compounds are found here close to the S2 helix, and 1 is on this side. Here again, the active site. To be mentioned is a zinc binding protease in parallel. So really complex and nasty, I would say, enzyme on this side. And here you see the ESG-15 binding site. We compared also to other structures already deposited. And you see here the 3 structures, again, I have to mention, obtained the compounds from Karachi. Here we have the names with well-defined electron density. 2 are here on this part and 1 is here. HE9 is our favorite today. They're well coordinated. So it's, and to prove that it's not an artifact, we performed ubiquitin assays to show, and they confirmed. I'm just showing again ESG-15 binding site here in the compounds. We compared also to other PLpros known from MERS and SARS Cov-1. So they have similar binding site, but not identical. So it is very clear that SARS Cov-2 evolved and is being more effective. You see here the comparison COV2, 1, MERS, and others They have all slight differences. And this is a clear evolution, which I'm worried, and scientists, we should be worried, humans, because the virus is developing further and also substantially suppressing the immune response. As just here is our assay here we are using to prove this. And we performed antiviral assays also. You will see here the 3 compounds suppressing substantial, this is a mutant non-active 100% here. And interesting, just to make it simple, this compound is also found in green tea, olive oil, and vanilla. So they are natural compounds. They have many applications for many years. And you have nice presentations from your colleague from China showing the history of natural compounds. These countries are very lucky. So in North Europe, where I'm sitting, our variety is much lower. And I finalized my presentation here. So with colleagues, we further analyze these compounds. They show the suppressive activity. These are tests we performed in hamster and mice, sorry. Here's the in vitro, and here is such the in vivo. We are just preparing one manuscript for submission, showing that one compound is super active and is suppressing the activity in mice and hamster very effective. And here's a summary of these compounds. We have many others in stock we have to look at. Many of you in the audience know these compounds most well and don't have to read again. They have applications. They are known to be antioxidant, anti-arrhythmic, so they're used to treat heart deficiencies. Again, anti-tumor compound, and again, this compound, which is today our favorite, we call it HE9. So both we going further to check these compounds. They are doing all these experiments I mentioned with mice and hamster. Here in Hamburg, we have the Institute for Tropical Medicine. There are also courses, also have the S3. You have to do this in a biosafety S3 laboratory. And we are doing this in parallel. I'm doing this also sometimes myself because I'm very interested at the Institute of Parasitology in the Sao Paulo University. I'm sorry, visiting professor here. I have a small office on this side. And because of time, I have to finish. But I need to acknowledge the collaborations, the Alchemains group on the DAISY campus, Henry Chapman groups, my colleague very valuable in interpretation of electron densities, Winfried Hinrichs, our friend and collaborators in the Karachi Institute, Professor Chaudhary and Dr. Attia, bioinformatic groups, and Fraunhofer, and my second home, the University of Sao Paulo, funding. And I use this picture because it's an old group picture without mask. It was just before Corona. Madhya Benish from Pakistan here. Maybe Malik is in the audience. He was a welcome visitor and colleague in our group. And here, Hina and Najib, they just obtained their PhD, and they are co-authors of all papers with Corona, and they just went back to their home university in Malta. I would spend 1 second. We have a workshop I'm organizing this June, again, will be hybrid. We are introducing all the methods in advanced biophysics, structural biology. We have very famous speakers on this side. We have tutorials in the afternoon. And I will forward this information to my colleagues in Karachi. So whoever is interested in your country to join these lectures or join even in person, they are most welcome to join this event. I have to thank the collaborators, HEC in Pakistan, supporting my PhD students from Pakistan, and the HEJ Center supporting the data bank. And I have to mention, we continue these efforts, and we hope that we can collaborate also in future, and of course, funding agencies. So thank you for following my presentation. maybe was a little bit squeezed with information, but I'm happy to present more details. And I mentioned I was planning to join the workshop in person, but as I was facing a high titer of COVID at the end of January and not passing the antigen test properly, I was in home quarantine. I decided to postpone and see you in the future in person. Thank you for listening.
Thank you, Professor Bezel, for such a wonderful talk on this topic. It's mind-boggling where crystallography has gone from where to where in the last 20 years in the area of protein crystallography. So wonderful talk. I believe we have 1 or 2 minutes for 1 or 2 interesting questions from the audience. They're all speechless. Wonderful. Thank you so much. Thank you. Exactly on time. We start the next lecture. I feel like going back to crystallography in proteins area now. I've been retired for 7 years. Such an interesting 7 seconds, 30 seconds you have data. Amazing.
It's free of crystalline substance.
Yeah. It's amazing one. We have a synchrotron in our neighborhood in Saskatchewan.
I think the major development in X-ray crystallography was, it actually went out of laboratory and all this huge wide radiation, very high energy. Synchrotron radiation has been a great find. And now data and other, while this not really happened in animals. So.
Okay, can you hear me?
Are we ready?
Okay,
Okay, please. The next speaker in our series is Professor Wei Sun, Institute of Chinese Bacteria Medica, Chinese Academy of Chinese Medical Sciences. And he will give us a talk on the omics study provide insight of routine accumulation in tartrate, buckwheat, seed, and sprout. He is very much interested in uncovering nature product synthesis in plants. Professor Wei Sun.
Okay, I would like to thank all the organizers for giving me this opportunity to introduce the work carried out in my laboratories in recent years. I came from the Institute of Chinese Material Medica, China Academy of Chinese Medical Sciences. This is my email. Okay. Firstly, I will introduce our institute and our location. Our institute located in Beijing and is very close to Forbidden City, and all the staffs in our institute are major in traditional Chinese medicines. The famous scientist in our institute is Professor Tu Yu. She discovers artemisins. Okay, I will introduce some work in my lab. Plants can produce a variety of small molecular compounds to regulate their growth and development and help them to resisting Baltico and albaltico stress. Many of these small molecular compounds have some medicinal properties for humans. When these integrants enter the human mouths, they reflect often bitter and sweet taste. There is an old Chinese saying that a good medicine tastes bitter and no sweet without bitter. Therefore, in my life, my interest is to understand the formation, the molecular formation mechanism of the bitter substance and the sweet substance. There are many plants can produce bitter integrants, including vegetables, fruits, nuts, and medicine plants. These plants can produce a variety of the compounds taste bitter. For example, some gentian roots contain some of the key integrants, bitter compounds called amylogentin, and the bitter compounds extracted from the citrus fruit peels include naphthene, is a flavonoid compound. And another example, just like the diserpinoid, the caffeine also tastes bitter. In my life, I'm focused on a special bison plant, or called a crop. The wild plant is distributed in China, India, Nepal, and Pakistan. This plant called Tartary buckwheat. In my life, we focus on this plant. These Tartary buckwheat seed and seedling can be used for the food, medicines. This plant is different from another crop, just like wheat and rice. It's a dicotyledonous plant. In China, many production can be used from the Tartary buckwheat. Two active integrants in Tartary buckwheat or bitter buckwheat. Number one is a flavonoid rutin. Another compound called isoquinine is another compound called imidine is isoquinine. So in my life, I focus on the quantification and the biosynthesis these compounds produced in Tartary buckwheat. So we want to answer some questions. So we use a different strategy to answer the three scientific questions or problems. What are the integrants in the bitter buckwheat seeds and seedling? The number two is what is the composition difference of the different Tartary buckwheat varieties? The number three is how are the active components in Tartary buckwheat produced and regulated? Next, I will introduce 4 papers in my lab in the 3 years. The first one, we want to answer these questions. What's composition of the tetrabackward seed. We use UPLC cutoff mass help us to identify these compounds in the seed. A total of the 71 flavanols and isoquinines were identified by cutoff combined with the reference materials and the literary comparisons. It includes 43 flavanols, 11 flavanols, and 6 other flavanols, and 11 isoquinines. These compounds these compounds are the main form of the glycoside form. Next, I will introduce why we study the glycoside of the flavanol and isoquinones. We also use the LC mass to optimize the extracted iron chromogram of the 6 flavanol and 11 isoquinones in tetrabackward growth. All the 71 compounds were analyzed in the positive iron mode, except for 10 compounds. When we finished the quantification and the qualification of the compounds in tetrabackward, we will answer our questions, the morphological characteristics of different germ plants of tetrabackward cultivars. We divided all the tetrabackward into the different shape, just like black, yellow, brown, based on the color, and also we also discriminate the tetrabackward based on the shape, just long and short. So next, we will use the above mentioned the metabolomics tools to discriminate.
Sofian, we cannot hear you. Sofian,
Hello?
we cannot hear you.
Oh, you cannot hear him? I thought I dropped, so actually he dropped.
Okay.
Okay, I call him.
Professor Wei, we cannot hear your voice. Professor Wei, your speaker is mute now. Your mic is mute. Dr. Wei,
He's trying to join again.
okay. Okay, we wait.
Dr. Swain, are you there? We cannot hear you.
He is online, but still not sharing his screen.
Can you share your slide and say something?
Okay.
Can you share your slide? We are not able to see the slide. Need share again.
Okay. Can you hear me?
Yes, your voice is clear.
Okay. We also observed the difference of the metabolomics between the difference of the tartar buckwheat sprout. We use the metabolomics tools to discriminate the different color of the tartar buckwheat. We also use the UVB to promote the flavanone and the isoquinones composition of the tartar buckwheat seed. The second question is why how the tartar buckwheat can produce the rutin and isoquinone compound. Glucoside is a final form of the rutin and imodin in tartar buckwheat. This glucoside can determine their pharmaceutical functions. Since imodin possesses very low solubility, and the practical potency as oral medicine will be discounted compared with their theoretical biological activities. So in my lab, I'm focused on the which genes determine the routing and imodine glycosylation. The imodine exists 3 hydro-distributed in 1, 6, and 8 position, which will be the target of the glycosylation. And the routing is main flavonoid in tartar buckwheat. They also contain the glycosylation and ramosylation. So next, we analyze the tartar buck genomes and use the genome sequence. With help of the genome sequence, we annotate the UGT glucose transference number. Total 106 UGT genes were found in the tartar buckwheat genome. These UGT proteins encoded by these genes are comprised of 300 to 600 amino acids. They have a significant PSPG motif near their carbon terminals. In my lab, we also found 38 of the UGT genes contains no introns. Next, we analyze these genes expression patterns using the transcriptome of the tartar buckwheat. We found among them 20 UGT genes possess high expression levels in the late developmental stage of the seed. Other 31 UGT genes were highly expressed at the stage of the ovary bulge. Finally, we want to determine which genes involved in the which UGT genes involved in the imodine glycosylation and routing glycosylation. We clone some UGT genes using the PCR, and we also using the enzymatic test to reflect which genes involved in the glycosylation. We found 5 UGT genes can use UDP glucose as a sugar donor towards imodine and quercetin. We also use enzymatic characteristic. We also show the enzymatic characteristics of the important UGT genes. We called tartar buckwheat UGT 33B5. We can catalyze the steps of the glycosylation of the quercetin and the imodine. In order to reflect the tartar buckwheat plant, the genes in the tartar buckwheat plant, we use a transgenic method to overexpression and RNA these genes in tartar buckwheat, and we use the LC-mass and to reflect the compound concentration. The tartar buckwheat transgenic hair roots can, the result can tell us the UGT1, also called 33B5, can determine the rooting and emoding production in Tatar buckwheat. The next question is what factors determine regulate the rooting compound and emoding compound. We use the seedling as model to study this, the regulation of the rooting. We use different light, LED light to to light the seedling. You can see the dark, the blue light, far right, and the right light. The seedling under the different light. We use the LC mice to determine the different light, regulate the compounds accumulation. We found blue light is a key factor to regulate the accumulation of the flavanols. We also observe the gene expression patterns in flavanode under the different lights. We use the qPCR to reflect the key gene expressions distributed in the flavanode pathways. We find
Dr. Wei, your voice is not coming again. Dr. Yan, we lost the voice of Dr. Wei.
Dr. Swain, we are not able to hear you again. Maybe you need drawing again the link.
Okay, so we have 2 minutes to conclude his presentation.
Okay, let me inform him. Dr. Sunway, can you rejoin? Yeah, he's trying to rejoin.
share this story.
Dr. Yan, can we go to the next speaker?
Yeah, just to let me confirm from him. Yes, he said, please go to next.
Well, I think we appreciate your efforts in communication around these things. But unfortunately, things the way they are, I would suggest to the audience, if you have any question from Professor Wai's son, his email address is given on page 12 of the program. So please communicate with him directly, ask him any questions if you have, and as we're going to move on to the next speaker. Professor Liang Kao is assistant professor at Hunan Provincial Field Laboratory of Dong Medicine, Hunan University of Medicine. He had an excellent PhD dissertation in Hunan Province, and in science and technology, the achievement of Hunan Province, we are public People's Republic of China award and honors. So I would request Professor Leong to please get your talk started. We have 20 minutes for this lecture. Please welcome Dr. Kao.
Thank you. Thanks for the introduction from the hoster.
Please switch on your camera and go for the full screen mode of presentation.
Yeah. The video may be not so quick to join. Okay. Can you hear me?
Yes, loudly and clearly.
Okay. Thanks.
Please maximize your presentation to the full screen mode.
Okay. So you can see the PPT.
Okay, go ahead.
Okay, thanks. Firstly, good morning, and assalamu alaikum to my Pakistani friends.
Dr. Liang, we cannot hear you.
Dr. Cha, we cannot hear you clearly. Actually, no voice at all.
Dr. Liang, if there is an internet issue, please switch off your camera so it may work.
Hello, Dr. Cha. Hello, Dr. Cha.
Dr. Yan, if the problem is still there, so we can switch his talk with another one who is ready.
Yeah, I'm calling him.
哎，王师，这个怎么回事哈？我一点开这个界面，我这里是非静音状态呀，怎么没声音呢？
Yes, your voice is not coming.
哎 ，Can you hear my voice?
Yes, we
Okay.
can hear you now.
Okay,
Go ahead.
Let's continue. Yeah, thank you. So maybe we turn on the video. The internet is not so quick, so
It's fine.
I close. Yeah. Okay.
Maximize your presentation and go ahead.
Okay. So this is Hunan province, and in the west of Hunan province, there are a lot of minorities people live in this place. Among Guizhou, Guangxi, and Hunan province, the Tujia and the Dong minority, the population is in top 10 in China. So we are interested in the herbal medicine in these minorities. Oh,
Please go to the full screen mode of presentation.
first one.
Full screen mode of the presentation. Yes. Okay.
Okay.
Go ahead.
Okay. So this is my university. It's an introduction of my university and the minority as medicine. So firstly, let's start with the Tujia people and the Tujia herbal medicine. This is a book from the Tujia people. Its main is investigation and clinical study on Tujia medicine in Western Hunan. And this old lady is a doctor in the Tujia minority. So my work is on one kind of herbal medicine from Tujia ethnic medicine. Its name is Xuetong. This plant, this Xuetong, is the stems of cultural heticulator, which are commonly used to active blood and relieve pain in the minorities, and also used to treat rheumatoid arthritis. Cultural heticulator belongs to Schizandra family. As we can see, this is the fruit of the plant, and these are stems. We collect the stem from the forest. It's very long and very thick. So the natural products from Schizandra, there are only two genus, Kajura and Schizandra from this family of Schizandra. And the plants in this family is high content in terpenoids, lignans, and flavonoids. In the recent years, there are nearly 500 new terpenoids have been reported from this family, and they all belong to the lanstan, cyclodine, and sphenoterpenoids. Sphenoterpenoids is very special because in the skeleton, the carbon number is 26 to 29, with carbon 18 or carbon 28. It's decarboxylation lost with the character with some compounds, some carbon lost in the structure, in the skeleton. So this is the structure, the core structure of the these classes of compounds. The chemical shift of oxygenated quaternary carbon, carbon 10. This carbon is in chemical shift of 96 to 100. And this signal is very characteristic for the sphenoterpenoids. So we are taking this characteristic NMR signal to track the SNTS in this in my plant shuttle cultural heat creator. And also SNTS have good bioactivity. This is the NMR common NMR spectra of the fractions from the plant of shuttle. We can see the top one. There are signals in the chemical shift of 95 to 100, but in another fraction there are no signals, these characteristic signals at all. So these fractions have these SNTS signals are our major our target to isolate the compound. Like from here, we isolated the non-terpenoids from the mixture, which the mixture contains some signals from SNTS. So this is carbon spectral gas isolation. And also we use prepared HPLC to isolate the ambutyl fraction of shuttle. As we can see from the picture, the two spots was eluted from the prepared HPLC. When we use TLC to check again, we can see these two spots were not separated. So we use semi-prepared HPLC to do the isolation with a different solvent system. And finally, we got the two compounds separated. And then one of them is have a beautiful crystal. So we come to illustrate the structures. We use both HRESI mass and proton, carbon, also the 2D carbons, 2D correlations, QC and HNBC to get the planar structure, and also Royce spectrum to determine the relative configuration. So this is the summary of the COSI and HNBC correlations of compound 1 and 2. From the results, we found that compound 2 is a carbon 5 apimer of carbon 1, because we can see the Royce correlation from this carbon 1 to carbon 5. So, and also we use ECD calculation to determine the absolute structure of compound 1 and compound 2, and along with crystal isolation, X-ray analysis use the Cooper solid target. From the stains of shuttle, we isolated some new terpenoids, new sphenoterpenoids, and sesquiterpenes. So this is the new sphenoterpenoids isolated from the stain of shuttle. This work was published in Phytochemistry in 2020. This 1, 2, 3 is precursor of sphenoterpenoids, because they are all, these three compounds have 30 atoms, 30 carbon atoms, but 4, 5, 6 are sphenoterpenoids. And also some known sphenoterpenoids was isolated from the plants. After that, we also isolated some other terpenoids, which is 3 and 3, 4 and 9, 10 dicycle cycloalkanes. This class of compounds we named them 血统素. This is published in Phytotherapy. The previous two work was the compounds isolated from ambutyl fraction. From the chloro fraction, we also isolated some compounds, some new compounds. This work was done by Nuzet Scheller, and she is a vice scholar from Karachi University. these new compounds were also cyclooctane star and land Xue Su. Beyond the triterpenoids, we also isolated some sesquiterpenes. This is 3 cardinal sesquiterpenes isolated from the Xue. As a conclusion of this study on Xue, so the phytochemical investigation of the scenes of cultural integrator, that means Xue, results in the isolation of structure. Results in the isolation of 21 new compounds, and among the known compounds, we found one compound named the cytoclysine H. These compounds have strong antioxidant effects, but this class of compounds is majorly from the microbes. So we think that the stems of Xue, because they grow in the forest for many years, so there may be microbes exist in the stem. That's why we isolated the compound from Xue. And also, Xue Su have good activity in anti-arthritis, in anti-inflammatory. So we got a publication on the bioactivity of Xue Su in rheumatoid arthritis. Another minority medicine as medicine is Dong minority and the herb medicine I wanted to introduce today. And Dong minority is a group of people very good at dancing and singing, and they are featured with beautiful towers and bridges built from wood. They have their own doctors, and they have the special detecting way to treat the patient. The doctors in Dong minority, they are performed to collect raw material, the raw herbal medicine to prescribe for the patient. We researched two kinds of Eastern medicine from the Dong minority. One is modern eye. We showed it for MDA. The plant source of MDA is potential Free-Electron. This is a small plant. The reason of the plant is used as modern eye. In literature survey, we found that in western of our Hunan province, there are almost 10 kinds of species was used in medicine, like potential Chinese. Most of these plants were used to treat dysentery, abdominal pain, diarrhea, enteritis, and menorrhea. All these disorders are related with infectious. So we think that this plant maybe have good anti-infectious activity. So we also do some literature survey and found out that the plants from genus of potential, most of them have good activity in have good antibacterial activity and anti-fungal activity, such as anti-SA and E. coli. So we use the water extract of modern eye to treat the MRSA and to treat the infectious. And the water extract of modern eye have good antibacterial activity, like anti-MRSA and anti-E. coli. After this work, we go to the isolation work for modern eye, and we use 95% of ethanol to extract the material, to extract the resin of the plant, and got it extract, and then we use patient ether, DCM, and ether acetate to fraction the extract. The bioassay found out that the ether acetate fraction from the plant, from the resin of the plant, have good activity in anti-SA anti-staphylococcus aureus. But in E. coli, there are no good activity. So the isolation work was done.
Oh, I'm sorry for interruption. Can you please conclude your talk within the next 2 minutes? We are running out of time.
Oh, thanks. Okay. So the work on this plant modern eye, we isolated some new compounds like lenic terpenoids and ozonic terpenoids. This work was done in Hunan University of Traditional Medicine. Another one is CSE, Cayatera albifolia. This plant have good anti-inflammatory activity, and the two work have been done. Use SPS and Zomus A, the induced inflammatory. The plant show curia activity. Also, the compounds isolated from the plants were phenoloids. So in our work, we just researched some kinds of plants from the minority medical system. And we found out that like modern eye and CSE have good activity. We are looking forward to further understanding the both the medicine system and the active compounds. So I'd like to to show my thanks to my institute, the Hunan Provincial Key Laboratory of Thermal Medicine, in this building, and Professor Zai Qizhang, and he is the director of our research institute. Also, I'd like to thank Professor Wei Wang. He's my PhD supervisor, and also Professor Arthur Iqbal and Professor Qingfu He. He is the president of our university. And also, I'd like thanks to the staff and the visitor scholars in HEJ when I was studying there in 2014 to 2015. And also, I'd like to thank Professor Lee Sinkong Li and Professor Ke Han from University of Mississippi. Okay, thanks for your attention. Thanks. 谢谢.
For the requesting lecture. Unfortunately, we do not have too much time for this. For any questions, his email address is given on page 12 of the abstract book. Please communicate with him directly if you have any questions. Thank you so much. And now we move on to the next speaker. Professor Kong Liu is assistant professor at the Institute of Food Science and Technology, Chinese Academy of Agricultural Sciences. Her interest is in neuropsychopharmacology, research on natural products, and she has an excellent postdoctoral fellow in Chinese Academy of Agricultural Sciences in 2018. Please welcome Professor Kong Liu.
Professor Lu, unmute
Okay.
your, share your presentation and go ahead.
Okay. Good morning, everyone. It's my great pleasure to attend this meeting. And my report is research and development of the function fold for neuromodulation. And I'll introduce 3 parts about research and development backgrounds of it, progress of our team's work in this area, and will predict the future direction of the function fold for it. As we all know, the neurodegeneration is that can't be ignored in the aging area, such as stroke, Alzheimer's disease, Parkinson, anxiety, depression, and so on. Heritage environment entering and other factors caused the gradual loss of the structure of the function of neurons in the brain, finally resulted these diseases. And the incidence rate is high and the harm is carried forward. According to the WHO, the neurodegenerative diseases are the hiding cause of disability, and it's practiced that it will replace cancer as the second leading cause death by 2014. And the endowments are complete and the pathogenesis is still unknown. Take Alzheimer's, for example, the A-beta hypothesis, the genetic hypothesis and trophizations are the main reasons in the current. However, the regulation size of key products in AD still needs to be identified, and such as in the progress of deprivation, the neurotransmitter hypothesis, the neurotraffic hypothesis, and attributable negative feedback disorders concludes the main factors in this disease. Well, the metabolic pathways and the key factors remains to be identified. In this situation, effective traits like and dietary nutrition can protect new ideas. According to the lesson report, diet is the second influence factors, about 13%, and the healthy consists about 15% in human health. Well, dietary nutrition intervention can protect the new ideas in the current. As an effective character research and development of functional food for neuromodulation is great significance. Then I'll mainly introduce the progress of our team's work in this area. Our team focused on food, nutrition, and functional factor utilization, and it consists of over 100 staff, including core matters, 4 professors, assistant professors, postdoctors, and master doctor students, and so on. The research of our team mainly about 3 effects. Firstly, we establish both the safety and the functional evaluation system for functional food. Then we describe the functional factors and clarify the measurements of their actions. Finally, we research on the efficient utilization of key technologies of the functional factor, and then developed the products, especially for the functional food. We have set up the related research platform. In this area, we collaboration with the research group of Professor Liu Xinming from Chinese Academy of Medical Sciences. Firstly, we developed several platforms, such as we have monitor mice tests, the object recognize device, the passive advice device for evaluating the cognitive function. Then we also conducted the Taylor suspension apparatus, the first swimming test, the learner helplessness test for assessing the anti-depression, the anti-anxiety effects. Also, we can take the open field tests direct for a sleep rate of animals for evaluating the sedative effects. And we also conducted the cell test postmortem and zebrafish test postmortem for the squaring the functional factors. Then we conducted several animal and cell models for neurological dysfunctions, such as the scopolamine induced classic monic ultimax mass model, the cortical depression mass, the cortical answer induced cognitive impairment in mice. Then we also conducted several depression animal models, both in rat or mice, such as cortical resistant stress and cortical unpractical space induced depression. Also, we conducted several cell injuring models in BV2 cells or PCC4 cells. Then we established the platform for the functional factor utilization and plot research for its steady state mice UC-fed use to create a series of functional foods. In the research on the theory and technology of functional food, firstly, we have scribe and font as one highly active sebum as a flower. Equal this actor shows the cognitive improving and antidepressant activities, both in several animal behaviors. And we have synthesis is mechanisms is cognition improving mainly through promote the cerebral neurotransmission, enhancing anti-oxidant system, inhibiting neural inflammations, and improving synaptic plastic. And its main antidepressant pathway is associated with an F-cannabis single path in the hippocampus, and its target is the BDNF site in the hippocampus of the mice. Then we separate analysis and ascribed one of active ingredient from a traditional Chinese product. Her Chinese name is Huanghua Cai. We found that flavorness are the effective compounds in it, showing the antidepressant factor, and the mechanisms showed that it's mainly by regulate the metabolism of tryptophan, glutamate, and energy. Then we analyze the key genes regulate the differential expression of flavors in different Huanghua Cai. Is about 4,3H sites, DFR sites, and C3H sites. Also, we further confirmed that polysites from one traditional Chinese medicine, Huangjin, exerts remarkable antidepressant actions by acute behavioral despair mice, critical unpredictable mild stress induced depression, both in mice, and its action mainly ensured by upregulating minor neurotransmitters, inhibiting neural inflammation responses in the brain. Then we also found one traditional Chinese medicine, 石斛 shows great effects against brain nerve injury, and the relation between structure and active showed that for C4 could match an important modification site in the structure of 石斛. Then, after research this, we manufactured a series of functional food for neuromodulation. For example, the milk powder rich, the different flavors, Cuban yogurt, and authorization as the sedative function, the bubble wort, the oral burst bees, chocolate, and other foods. Finally, we'll predict the future direction of the functional food for neuromodulation. The firstly, is the product function will be balanced. Adopt the new research approach of making the more matters and adding different factors to make the products more effective and break through the traditional way of using one single neural function integrated, and finally realize the transformation from single nutrition to balanced nutrition. Secondly, the product form will be diversified, including the dominant form such as capsule, soft, sandy beverage. More convenient product forms such as lithium food and trolling gum will continue to emerge to make them more diversified to meet the daily needs of prevent the diseases. Thirdly, the product orientation will be personalized. For example, in developing the sleep development function food for women, we can combine the oral beauty elements to meet personalized precise nutrition and health needs. It will realize the products more targeted. First, the product formats will modernization, realizing the China's rich food and medicine raw material sources and the long history of medical foods. Integrate the traditional Chinese food theory collector, modern food products technology, and biology methods to achieve the modernization of functional foods. Okay, that's all. Thanks very much for all of your attention. Welcome to our lab.
Thank you, Professor, for the interesting talk. I think we have a couple of minutes for questions, if the audience have any questions. Any questions? Yes.
That was indeed a very interesting talk. But I have a question that how this functional food with the spatial ingredient added will be different from the drugs available in the market. For example, in one of the slides you have shown that one of the products will have the added GABA in it. So how will it will be different from the already available drugs which are available in the market? to modulate the GABA function. Did you hear my question?
Hello, Dr. Lu, did you hear the question clearly?
Sorry, I can't hear clearly.
Yeah, actually, I also not able to listen clearly. The voice a little bit mixed with some
Can you hear
noise.
now? Can you hear now?
Yeah.
Okay, my question is that how your functional food with the special added ingredient in it, for example, one of the food you have seen with the added GABA in it, how it will be different from the available drugs in the market. For example, for the GABA modulation, you already have the drug. So how it will be different from the functional food?
Sorry, I also didn't get.
I believe that's the end of the session. Thank you everyone so much. There are short announcements about the tea break. The next session will start at 11:25. Please note that there is a slight change. Both parallel sessions are now arranged in LEJ Hall A and B. There will be no more lectures in this room anymore, I think. So please go to the LEJ building. Hall A and B will be used for the rest of the lecture.